So, okay, yeah, I am. So, good day to everybody. And first of all, I wish to thank all organizers and colleagues to have invited me to this outstanding event. Really, really informative. Also wish to apologize for not being attending in presence for several duties. Indeed, I'm very and deeply grateful to grant to me the opportunity to join also virtually. So honored to be with you and here to network, to interact to you all on these very interesting themes, basically genomics and advances in genomics about rare diseases. So for the next slide, please. So that these are my disclosures. So just to mention briefly that I'm also a PI of some clinical trials for Duchenne muscular dystrophy, which are sponsored by PTC Therapeutics and Sarepta Therapeutics. The next, thank you. I put this slide as a first, just to show you an overview about what is going on in Europe for rare diseases networking and initiatives. So we know the prevalence of rare diseases. So Dr. Amal has so nicely basically introduced the argument of the genetic and genomic diagnosis of rare diseases. So in Italy, the European Union has established the so-called European reference networks. These are, a network of healthcare providers across all countries of the European Union, which are in charge of rare diseases care, diagnosis, and therapy. And you see here that in Europe, we estimate to have about 30 million of patients belonging to these 24 different phenotypic categories, which are also the expertise, so the duty and the main characteristics of each ERN. So basically we have, you see here the list of ERN below, ERN for specific phenotypes, so neuromuscular diseases, rare neurological diseases, intellectual disabilities, and so on, in these many types. And this is the report, is a slide, kindly provided by the DG Santé in EU, which is, you see here how many patients are now followed up by each ERN. And this is also reflects somehow the number of rare diseases which belong to the specific phenotype category. So this was a great implementation in Europe. And the main improvement, the main impact was to have, for the first time, all the reference centers, so centers in charge of rare diseases, connected in a network and then interacting, also using digital tools. The next. So we know, because you have already presented, some colleagues, clues about the next generation sequencing. So indeed, this was really a revolution in medical genetics. I'm a medical geneticist, so this was really challenging in the last, let's say, 15 years. So now what we can do in terms of technical strategy, we can sequence single genes, so targeted sequencing. You see that on the right. So basically, you enrich your sample for including specific genes of interest, and you go to sequence those genes. Basically, mainly only in the coding regions, so on the exons. This is very common, known as gene panel, so a sort of panel including many genes, which we are going to sequence, and also to identify other mutation types as copy number variations. That's a very important point, to have a very high detection rate and to have a very accurate diagnostic pathway. Or we can use whole exome sequencing. There are many types also available as kits from companies. And you see that using that approach, you are going to sequence all exons in our human genome. So coding regions as well. That's, of course, much more comprehensive, and you may be able to sequence ideally all genes underlying rare diseases. The last one, which is very attracting, of course, is whole genome sequencing. Now we have also, a third generation of whole genome sequencing, so-called long reads whole genome sequences, which is able to sequence the full genome of an individual and also to pick up non-coding regions or regulatory regions, intronic regions, and so on, and to see quite nicely also small variants, but also copy number variations, and is in progress the possibility to see also triplet diseases or dynamic mutations, which escape completely now the possibility to be identified by using gene panel or also using whole exome sequencing. The next, please. We have issues which are addressed and are becoming to be addressed quite a lot now. So obviously, sequencing West and sequencing whole genome, we have a huge, we say, output, number of data, number of reads in nucleotides and sequences. So we have to manage how to go into detail and to interpret this output and variant interpretation, as also Dr. Amal has mentioned it before. We have guidelines. Also, we have seen that in the first nice presentation today for the American College of Medical Genetics to address the variant interpretation using mainly in silico metrics. And of course, we should improve our capability to detect all variation types. That's important for a medical geneticist, and also to be sure that you have really achieved your diagnostic goal, so to have really the gene fully analyzed in terms of mutation types. You see here the different in terms of base targeting and also in the output that you can have in using the different approaches. So whole genome, West, large and small panels, which implies, of course, a lot of filtering activity and output analysis activity. The other great added value of using next generation sequencing as omics is to identify other inheritance models, as for example, digenic inheritance. That's very common now when you analyze, for example, West data or even big panel data or genome data. You may find digenic inheritance due to different mutations in different genes. That is something that was really neglected in the past using a single gene approach. Mosaicisms are also very well identified by NGS because you have this very in-depth coverage, so many times you read a single stretch, a single region of sequence, and also the very important model of mutation load, which is basically not only digenic inheritance model, but you're including more genes. So in a sort of polygenic model, with a master gene playing a major role in the disease onset, but also orchestrated by the contribute of other genes, which are also mutated in the same individual. The next, thanks. Very quickly, in the routine diagnostics that we are involved in, all of us doing, providing genetic diagnosis to our rare disease patients, we are still using old techniques. So, and these techniques may be adaptable depending on the main question we have in terms of diagnostic, and also depending on the diagnostic accuracy, diagnostic grade. So definitely single gene sequencing is widely adopted for prenatal testing, just for an example, and still for dynamic mutations now, and still for epigenetic changes. But also we can use panels that are very popular, very largely used in diagnostic setting, for, diseases with very similar phenotypes. So, for example, the cardiogenetics panel, the neuromuscular panel, the intellectual disability panels, these can be very facilitating diagnosis, and also that should be added and underlined. They may have a very short turnaround timing. And obviously, you can use WES and whole genome sequencing for all other situations where genes, for example, are not known, so gene discovery task, or high genetic heterogeneity, or phenocopies, so very not homogeneous phenotypes with the underlying genetic defects. So that's then can be used, all together or in different steps in the diagnostic setting. The next. So summarizing, we have NGS activities and methods and single gene testing, which are providing a different in-depth diagnostics. And so we can be flexible and use all of them in different situations for, specific cases which should be addressed, of course, to provide the final genetic diagnosis to our patients. As you see, there are some, red faces, meaning that for some, for example, dynamic mutation epigenetic changes, high throughput techniques are still not very good in terms of detection rate and capability to pick up defects. So, but for sure, methods will be great in a few years and will be improved. The next, please. I was involved just to show you that we are working also already on whole genome sequencing guidelines for diagnostics. So we have now some orientations on how to apply and to translate whole genome sequencing in diagnostics facilitated by the European Society of Human Genetics. The next. And then the point is, where are we now? So, and the key point is that probably we are translating genomics from diagnosis to screening. This is the step that we may have to jump into the genomic health. So going from single individual diagnosis for our patients, our rare disease patients, toward a population screening, of course, different populations, cohorts that are widely analyzed in order to what? In order to prevent, to have an early diagnosis, or of course, and then to cure and to provide therapies to the patient. And the key milestones that we are already pursuing now in terms of getting the screening is, for example, population genomic screening, or pre-implantation genetic screening, non-invasive prenatal testing, and the last is genetic newborn screening. The next, please. Briefly, around all these steps, so NIPT, so non-invasive prenatal testing, has revolutionized the prenatal approach for rare disease or for couples at risk for rare diseases. So this is a fantastic technique able to analyze fetal DNA free circulating in the maternal blood. So, and the analysis of this DNA can be done using next generation sequencing. The next, this technique is now largely used, for example, for chromosomal abnormalities, because it's able to pick up copy numbers which are, of course, disclose the presence of trisomies, for example, or any other aneuploidy. So is very impactful because it's a non-invasive prenatal testing. The next, but not only, there are a few experiences now already published of the use of this fetal DNA circulating in the maternal blood to also get a prenatal diagnosis on specific gene or even on group of genes in terms of screening of genetic diseases at the prenatal level, at the prenatal age. That is also quite relevant and impactful in terms of providing a cohort of interventions for prenatal, at the prenatal stage. and so for couples at risk for genetic recessive or X-linked or even dominant genetic disorders. The next, please. Of course, I will go quickly on that because pre-implantation genetic strategies are also quite well known, and then they are based on the analysis of blastomers in course of in vitro fertilizations in order to identify mutated, so carry the mutations, blastomers, and to do a selection thanks to the PMA or, assisted medical fecundation. So this can be applied for screening also chromosomal abnormalities and also gene mutations as a panel genes, so used based on targeted sequencing approaches. The next. And then also preconception carrier screening was also mentioned by the speakers before myself as Dr. Amal, and that's also obviously quite impactful on quality of life because it allows couples to consider basically the most complete range of reproductive options. A pre-screening, preconception carrier screening can pick up carriers of rare diseases, heterozygous, of course, rare dominance with different penetrances, and then, of course, couples may know that they are at risk for one or more diseases, and they can plan also their reproductive choices, and including, pre-implantation testing or prenatal testing or even other possibilities. So that's also interesting. I reported two examples. The next slides. This is what was kindly provided to me by BGI. So the courtesy of Wenyan Li, they are doing in China a lot of pre-conceptional testing, screening for many, many genes. Examples here of 50, more than 100 genes on couples with, infertility burden. And this, of course, allowed to identify many genetic defects that, of course, were underlined and addressed, providing opportune, prevention or, genetic strategies or screening strategies to prevent or early diagnosing rare diseases. And the next, also similar to China, also Australia. The next, thank you, is also carrying on preconception carrier screening in 3 different countries. 9 studies were published in 2021 about the possibility to investigate gene panels in these couples to identify at the preconception stage genetic defects, and then to provide a very comprehensive, counseling and prevention and reproduction choices to the couple. So indeed, quite interesting data. The next slide. And the last, of course, just briefly introducing what is a screening concept that probably we should have introduced before, but never mind, please forgive me. So which is the difference between screening and, of course, diagnosis? When you screen a population, and the challenging point for the future now that perhaps would need discussion, networking, because this is also challenging for the genomic data management, is that you are screening population which is completely or might be completely healthy. So our approach is not for patients or for sure at-risk people, but for healthy people, in order to profile their genome, their genotypes, to identify who is carrying a specific genetic defect. And this goes directly to newborn screening, which is now challenging because it's going to be translated into genomic newborn screening. So not metabolic, but genomic newborn screening. There are many initiatives ongoing worldwide. I listed some of them, I know, and I'm aware, and I also involved in this worldwide group of study from United States and BabySec and many others. Also in Australia, also in China, and probably many others I'm not mentioning because I do not know. This is something that we have published together with Genomics England, for example. And the next, please. just to introduce you, I'm happy to have this very short, spot about the project I'm coordinating in the European Union, which is Screen for Care. I'm co-coordinating the project with Pfizer. Indeed, this is a new style of EU project, so where you have also many companies involved, which is Screen for Care. And Screen for Care is really piloting genetic human screening based on gene panel and also whole genome sequencing in a very wide population in Europe of about 25,000 infants. You see here Nicola and myself. The next, please. This is the large partnership of the Screen for Care, and you see that there are academics, there are companies involved in drugs development, but also in visual diagnostic. This is something that we are trying really to put together our expertises, our critical mass, and to build up a fully translatable model to be impacting on health of our children, of our population. The next, please, which is probably the last. So just the summary of Screen for Care. For you, if you like, please go on our website to know more about this project, which is ongoing. Duration is 5 years and is spread around Europe. The next. That's a very possible pipeline that is for discussion with you all. So, which includes all these figures of screening. Might be preconception screening, might be, of course, pre-implantation testing or newborn genetic screening or NIPT. That might be, more, let's say, appropriate or more usable, if I can use this word, apologize, for some specific situations related to rare conditions. They can be adapted to population screening or to early diagnosis and prevention, or to screen patients to get early therapies, for example, as the outstanding example of SMA that the colleague was presenting before, fantastic data about therapy, and also to prevent largely in couples. So using all of this, we should take into account about an outstanding aspect, which is ethics. Please, the next. And also not only ethics, but also the challenging issue to get toward the genomic medicine is privacy, is genomic data ownership. This is also very outstanding point to be faced, to be discussed. How get data access? How can we share data? How data are curated during time? Because we will have genomic data from people, from healthy people, from babies. So we should think on how to manage and to properly use this data in an ethical way. The next, I think, is just acknowledging you. Sorry for being a little bit longer. I'm happy to have your questions. Thank you so much again for having invited me today. Thank you.